So now let's have a look at synaptic weight changes from a chemical perspective. This shows the neuron soma and one synaptic spine located out in the dendritic tree with a glutamate synapse equipped with NMDA receptors. Inside the soma is the DNA surrounded by the nuclear membrane, and I've illustrated a little bit of the extension of the nuclear membrane out into the dendritic tree. This extension, it's called the endoplasmic reticulum, is present throughout the dendrite. Segments of DNA are activated by the activation of appropriate chemicals, and one example of such a chemical is Krebs. In this diagram there are dopamine receptors located close to the base of synaptic spines, and also dopamine receptors on the soma. The neurons that release dopamine to these synapses are all located in a structure called the midbrain dopamine neurons down in the brainstem. Then there are receptors for acetylcholine located on the trunk of the dendritic tree, and many of the neurons that generate this acetylcholine are located in a structure called the basal forebrain. Finally, there are voltage-gated calcium channels on the soma. So with this diagram, let's try to understand how very long-term synaptic weight changes are managed. Calcium entry through NMDA receptors at the synapse initiates a kinase cascade leading to activation of the kinase ERK2 close to the nucleus. Calcium entry through voltage-gated channels on the soma initiates a kinase cascade leading to activation of the kinase ERK1. Dopamine binding to receptors on the soma and at the synapse activate a pKa kinase cascade. And all these paths encourage activation of the DNA activation factor Krebs. So they can all encourage synthesis of the messenger RNA corresponding with actin. In addition, cholinergic M1-M3 receptors activate a pKc cascade. That pKc cascade leads to release of calcium from stores in the endoplasmic reticulum where it's kept isolated from the rest of the neuron, and this calcium activates the kinase CAMK2, which in turn activates other DNA transcription factors. So there are multiple interacting factors that determine whether and to what degree actin messenger RNA will be synthesized. Now let's go in and look at this in even more detail. Ultimately, neuron signals arriving outside the neuron membrane must trigger DNA transcription down in the nucleus. And that transcription must be initiated by transcription factors. One route to DNA transcription is initiated by the firing of dopaminergic neurons, which act on the D1 receptors to activate pKa via a second messenger. The protein kinase A can then activate a DNA transcription factor. A second route is by entry of calcium ions through NMDA receptors. This route leads to activation of the CAMK4 kinase. Within that route, the kinase CAMK2 can directly encourage DNA transcription. Then glutamate can also act via a G protein receptor, activating the MAP kinase via a different cascade. The same cascade can also be activated from the NMDA receptor. The glutamate mGluR5 G protein receptor can also act to release calcium second messenger molecules from the endoplasmic reticulum, affecting other cascades. Also, it can act via the protein kinase C to influence the calmodulin kinase cascade. So there are lots of parallel kinase routes to transcription of the same DNA segment. And I haven't even illustrated the acetylcholine-initiated route, and these are only the major known routes. There's also cross-connections between the routes. Some routes can inhibit other routes. And I haven't even mentioned the phosphatases that can deactivate kinases, partly because they're less well understood. So the management of whether the skeleton within a synapse will expand is very complex, with lots of cross-connected routes, and we need some way to come to grips with this complexity without being overwhelmed by it.